Hello everybody and welcome to your very first lecture of the course for microbiology. Today's lecture is just going to be an overview and possibly a lot of review from some previous biology courses you might have taken. And we will be going over some different classifications of microorganisms such as bacteria. And we will also be talking about just a little bit about the origin of bacteria and a general understanding of the origin of life. The learning objectives for this specific lecture, we're going to outline how organisms are classified. You might have heard of the term taxonomy. We'll be going into that more in detail. We're going to be able to list the main distinguishing features of archaea and bacteria. We will be able to compare cyanobacteria and protobacteria in terms of general features and metabolism. And most importantly, for this lecture, we will be able to describe the structural difference between gram-positive and gram-negative bacterial cells after we have stained them. After this lecture, you will be able to list examples of adaptations that have increased bacterial cell complexity throughout millions of years. You'll also be able to define terms such as autotroph, photoautotroph, chemolithoautotrophy, heterotroph, and photoheterotroph. You will also be able to define and understand the terms obligate aerobe, obligate anaerobe, aerotolerant anaerobes, and facultative anaerobes. So this might just be some review for some of you, but we're going to be going over the classifications of life. You might have heard of the term taxonomy in the past, and if you have, you know that taxonomy is the grouping of species based on common ancestry. There's 3 domains of life: bacteria, archaea, and eukarya. Bacteria and archaea are both prokaryotic unicellular organisms, where eukarya or eukaryotes are unicellular or multicellular. No matter what kind of life we're talking about, whether it's archaea, eukarya, bacteria, you, me, or your dog, when we're classifying organisms, we're using a system of taxonomy in which we classify life through species. In terms of taxonomy, species would be the most specific classification, and then genus. And both species and genus go together with something called binomial nomenclature, which we will be talking about a few slides from now. So, we know that human beings as a species evolved, all from a common ancestor. And so have every other living being on this planet, including bacteria and other microorganisms. So let's talk about microbial evolution for a minute. There's this amazing thing called horizontal gene transfer, which is increasingly recognized as an important evolutionary mechanism that increases genetic diversity among microorganisms. With horizontal gene transfer, at least 17% of genes present in E. coli bacteria came from other bacteria, and about 80% of prokaryotic genes have been involved in horizontal transfer at some point in their history. And this goes back millions and millions of years. But let's move on to binomial nomenclature, which is what I was just briefly talking about. So every species has a unique scientific name with a genus and species formatting. The genus part of the name is always capitalized, and the species descriptor is not capitalized, and the whole thing is italicized. So here's an example right here of E. coli. There is also an example here of Salmonella enterica. Salmonella being the genus and enterica being the species. Every living being that has been discovered on this planet will have a binomial nomenclature name with a genus and species. Now, microorganisms are typically so small that they need to be visualized with a microscope. One of the most prominent features of the microorganisms is their diversity. So, like we were talking about before, we have our prokaryotic domain archaea, prokaryotic domain bacteria, and in terms of microorganisms, we have our eukaryotic domain, which includes protists and fungi. If we look at the domain bacteria, we will see that it is mostly unicellular prokaryotes, and they inhabit many diverse environments on Earth. You will find bacteria on almost any surface anywhere on Earth. Now, if we're talking about archaea, they are also our unicellular prokaryotes, but they often live in extreme environments, such as hot springs. You will often hear them referred to as extremophiles, where they may favor really hot situations, really cold situations, very acidic situations, etc. Archaea and bacteria are so small, but yet they are the most abundant type of organisms on Earth. Did you know that half of Earth's biomass is bacteria and archaea? Archaea and bacteria are each monophyletic, which means they each have a single common ancestor that gave rise only to that group. So basically, that single common ancestor is what distinguishes these groups. Like I was saying before, archaea and bacteria live in nearly every conceivable habitat on Earth, including extremely hot, salty, chemical, things like that. Basically, they are able to live in places that do not support any other type of life. They have an extraordinarily high metabolic diversity. Therefore, they play diverse ecological roles, which is why they can live in environments that support no other life. They're usually single-celled, but some can aggregate into colonies or filaments. We will be getting into the evolution of life shortly, and we'll be talking about cyanobacteria, which formed colonies on the sea floor millions and millions of years ago. But for now, let's talk about extremophiles. So, extremophilic archaea and bacteria occupy habitats with extreme conditions. Halophiles are archaea which enjoy high salt content environments. Hypothermophiles enjoy environments that are of high temperatures. Here is an example of Thermus aquatus, which thrives in temperatures between 50 to 80 degrees Celsius. Currently, to this day, we know of at least 50 diverse bacterial phyla, with many still poorly understood. The phyla of bacteria is so diverse, it will take us thousands of years to study to fully understand. Cyanobacteria and Proteobacteria are especially diverse and extremely important, especially cyanobacteria. Most bacteria favor moderate conditions, but some are examples of extremophiles, and many form symbiotic relationships with eukaryotes. Now let's talk briefly about cyanobacteria, because cyanobacteria is extremely important to our existence. Without cyanobacteria, life on Earth would not exist the way it does today, or if even at all. So I keep saying cyanobacteria, but what the heck is cyanobacteria? Well, cyanobacteria are photosynthetic bacteria. They're abundant in freshwater, oceans, and wetlands and soil surfaces. They are named for their blue-green or cyan color. They are the only prokaryotes that generate oxygen. Now, if you're picking up what I'm laying down, you're thinking, oh, without cyanobacteria, there would not be oxygen on Earth. The emergence and colonization of cyanobacteria was so important for the evolution of life on Earth because they gave rise to photosynthetic organelles of eukaryotic algae and plants. So without cyanobacteria, there would be no plants on Earth. And without plants on Earth, there would be no oxygen. And without no oxygen, there would be no you. So of course there's different types of cyanobacteria, and this is what they look like in colonies. This is what unicellular cyanobacteria looks like. They can also be seen in unbranched filaments as well as branched filaments. So like we were just talking about, they played an essential ecological role in producing carbon and nitrogen, as well as oxygen. So what about protobacteria? Protobacteria do not produce oxygen, but they play other ecologically important roles. They come in 5 major classes: alpha, beta, gamma, delta, epsilon. They have incredible diversity of form and metabolism, and they include ancestors of mitochondria as well as human pathogens, such as gonorrhea. So how do bacteria survive harsh conditions? Well, bacteria have endospores, which encase the DNA within a tough protein coat. This keeps genetic material safe until conditions improved, and you can find them in some gram-positive bacteria. Now, bacteria come in a bunch of funky shapes. And of course, each shape has a scientific name. If a bacteria comes in a sphere shape, we call it cocci. Here is an example of what a cocci-shaped bacteria looks like. If a bacteria is rod-shaped, we call them bacilli. Here is an example of a rod-shaped bacilli bacteria. For the purpose of this course, we will not have to know about comma-shaped bacteria called vibrios. But for your reference, here is an example of vibrios. So we only need to know 3 major types for this course, 1 being sphere, 2 being rods, and 3 being spiral. And we call spiral-shaped bacteria spirilli. And here's an example of spiral-shaped spirilli bacteria. Let's talk about the structure of the cell walls of bacteria. So most bacteria have a rigid wall outside the plasma membrane that maintains the cell shape and provides protection. The cell wall also helps avoid lysis in hypotonic solutions. If you don't know what lysis is, it's the breakdown of the cell wall. Most bacterial cell walls are composed of peptidoglycan. Now, when we're in the laboratory and we're preparing slides, we stain our cells. And bacteria can stain either gram-positive or gram-negative. When we stain cells in the lab, when they are staining gram-positive or gram-negative, gram-positive bacteria will stain purple, and gram-negative bacteria will stain pink. And what is staining in the cells is actually that peptidoglycan cell wall. So, gram-positive cell walls. So, gram-positive cells have a relatively thick peptidoglycan layer. The dye is held in a thick layer that is stained purple. So, when we're talking about gram-positive, think thick and purple. But don't worry, we will be going and showing examples of what the slides will actually look like. And also remember that gram-positive bacteria are also vulnerable to penicillin, which interferes in cell wall synthesis. Now, gram-negative cell walls. There's less peptidoglycan and a thin outer envelope of lipopolysaccharides. So like I said before, gram-negative stains pink. And it will also be thinner. So when you're talking about gram-negative, think pink and thin. They are also able to resist penicillin, and they require other antibiotics. So they are penicillin-resistant bacteria. Now here is a visual example of what gram-stained bacteria will actually look like on a slide on the microscope. So on the left-hand side here is gram-positive bacteria. They are purple and the cell walls are thick. I mean, these aren't very good photos. It's much better when you can see it through the microscope yourself. But if you were to look very closely, the cell wall is very thick compared to the gram-negative bacteria, and they are purple. And then the gram-negative bacteria, the cell walls are stained very thin, and they are pink rather than purple. So, talking specifically about cells and their intracellular structures, some bacteria have complex intracellular structures similar to the organelles of eukaryotes. For example, cyanobacteria can use light energy, photosynthesis, because they contain thylakoids. We will explain thylakoids on the next slide. Magnetospirillum has magnetosomes that respond to Earth's magnetic field. We will also be talking about magnetosomes. And then planktomocytes have nucleus-like structures. So thylakoids provide greater surface area for chlorophyll and other molecules involved in photosynthesis. They are present in cyanobacteria and other photosynthetic bacteria. Ingrowths of the plasma membrane increase surface area and contain chlorophyll for photosynthesis. So magnetosomes contain crystals of magnetite which responds to the Earth's magnetic field. So in this photo right here, we have a photo of a bacteria with a flagellum, and inside the bacteria you can see this row of magnetosomes, and each magnetosome contains one magnetite particle. And they are enclosed by a membrane derived from the plasma membrane. Magnetosomes help orient the cells and survive in low oxygen habitats. Now let's talk about cell metabolism and the diversity of such. When we're talking about metabolism, there's several categories we need to go over. Autotrophs, which you might have heard of before, they produce all or most of their own organic compounds from inorganic sources. Photoautotrophs which is the most commonly heard of, use light as a source of energy. So you might be thinking photosynthesis. And you would be correct. So photoautotrophs are a type of autotroph that use photosynthesis to get their energy. So they make their own energy through light. Now, chemoautotrophs get their energy from inorganic compounds by nitrification or oxidation. So basically, chemoautotrophs make their own energy by synthesizing nitrogen and oxygen. And remember, both photoautotrophs and chemoautotrophs are types of autotrophs, which means they make their own energy. Now, heterotrophs require one or more organic compounds from their environment. Photoheterotrophs use light energy to generate ATP, which is adenosine triphosphate, but use organic compounds as a carbon source. Whereas chemoheterotrophs must use organic molecules for both energy and a carbon source. Also remember, heterotroph means they must take something from their environment in order to gain energy. They do not make their own energy, they need to get it from somewhere else. Now, bacteria do need oxygen, believe it or not. And there's different kinds of bacteria and their oxygen needs. So let's just go over a few of the most common here. Obligate aerobes require oxygen in order to survive. Without oxygen, obligate aerobes would die. Now, obligate anaerobes are poisoned by O2. So if they're in the presence of oxygen, they would die. And for the purpose of this course, we don't necessarily need to know about air-tolerant anaerobes. But for your reference, they do not use oxygen, but are not poisoned by it either. They obtain energy through fermentation. And the other one you do need to know is facultative anaerobes, which can shift between modes depending on conditions. We have been studying bacteria for hundreds of years. Bacteria are so useful in the environment, and they have an enormous impact on human health, good and bad. Remember, bacteria divide quickly by binary fission, which is an asexual method of reproduction. And this allows for bacteria to be grown quickly in a lab for study, which makes them easy to identify, characterize, and manipulate. When you are in the lab, there's several methods of visualizing bacteria. Now, of course, you can observe bacteria undergoing division, like binary fission, asexual reproduction, under a microscope. You can also visualize bacteria by growing them in an agar plate. Bacteria are also easily visualized when staining them with fluorescent dyes. There are so many ways to visualize bacteria, study them, manipulate them, you name it. When it comes down to it, bacteria are everywhere, they outnumber us, and we rely on them. There's bacteria in your digestive tract right now, helping you digest the sandwich you ate for lunch. There could also be bacteria on your hands right now from rubbing up on a surface that have the potential to make you sick. Now, to summarize the presentation for today, bacteria have been on this earth for millions of years. They've gone through evolutionary stages right alongside us, and they are the most abundant source of life on earth. Without bacteria, life on Earth would not exist. And that is the end of our lecture for today. If anybody has any questions, I welcome. The comment section is open. I will try to answer as many questions as I can.